All right. So in this lecture, what we're going to look at is we're going to look at the sequence of events that takes place during the neuronal action potential. All right. So when we're talking about the action potential, right? And if we go ahead and write this up here, the action potential is essentially just our electrical impulse that is going to be generated and propagated down our neurons, right? So all it is an electrical impulse. Okay, it's an electrical impulse, so it's electricity essentially that is going to travel down our neuron. Okay, so if you recall, our nervous system, we have two broad types of cells, right? We have neurons, which is what we're talking about here, and then we have glial cells as well, right? And so in this case, what we're talking about are our neurons. Okay, so if you recall, a neuron has a soma, right? So it has a cell body, it has a long kind of slender axon, it has dendrites that come into it, it has axon terminals at the end, right? Here would be a nucleus, and this action potential is going to travel down this axon toward the axon terminals. Okay, so we can graph and we can represent an action potential using let me draw a better graph here, using a nice graph like this. So what we're going to start off with, this is going to be time down here, and the specific time in milliseconds, and the specific time is not overly important. And then we also have voltage along this axis, specifically in millivolts, right? So what we have take place during our action potential are a couple of events, okay? To begin, we're going to actually plot or actually write a few important values here, okay? So one of these values is going to be, let me make this a little more bold here. One of these values is going to be negative 70 millivolts, roughly, okay? One of these is going to be negative 55 millivolts, right? And one of these up here, this is going to be, we're going to put zero somewhere in here, 0 millivolts. And then up here, we're going to put something like positive 30, okay? We're going to put positive 30 millivolts, okay? So what we have is our action potential, okay? So our action potential is always going to start at what's referred to as a resting membrane potential, okay? A resting membrane potential. What this means is at rest, before our action potential actually takes place, we're going to be hovering around a particular value, and this is going to be this negative 70. So this is referred to as the resting membrane potential right here. Okay, so this negative 70 is the resting membrane potential. So if we plot this, let's plot it in, let's just draw it in red here. We're going to be somewhere right in here, right? We're going to be existing and hanging out somewhere around negative 70. And if you recall, different ions that we have, sodium and potassium primarily, have what are called equilibrium potentials, right? So we have an equilibrium potential for sodium, we have an equilibrium potential for potassium, and they're very different from each other, okay? So each one exerts its own effect on the neuron and causes the neuron to move toward, when the membrane becomes permeable to a particular ion, it causes the resting membrane potential to move closer to the equilibrium potential of that particular ion, right? So for potassium, it's something like negative 90. For sodium, it is quite a bit more positive, somewhere around positive 60, okay? So what we have is we have our membrane more permeable to potassium, so our resting membrane potential is going to be closer to that equilibrium potential of potassium, which is somewhere around negative 90 or so, okay? Now, we also are slowly leaking sodium out through sodium leak channels, and we slowly kind of begin to move up kind of toward this next value of negative 55, okay? And this value of negative 55, this is what's termed our threshold. So let's write this in, threshold potential, okay? Is this negative 55 right here, okay? This is our threshold, all right? So we can also receive a stimulus. Okay, so there can be some stimulus that arrives, right? So let's write this. We can have some type of stimulus arrive that can further depolarize our neuron, right? So if you recall, depolarization is the membrane getting more positive, and really this stimulus would be a excitatory postsynaptic potential. So really this would be an excitatory postsynaptic potential, which is positive, right? Which is making us move closer to threshold, right? Which, and again, threshold is this -55 value that we have right here, and we're just going to draw this kind of going across like this. Okay, so if we have a sufficient enough stimulus that gets us up to threshold, or gets us up to our -55 millivolt threshold, and we're at that threshold at the axon hillock, right? So remember, the axon hillock, if we look up at our diagram here, the axon hillock is going to be this area right in here, this kind of central region. Sometimes it's called the trigger zone. That is where the action potential is initiated. That's where it begins. That's where it starts, okay? And that's the only place it can start. So you have to be at threshold right here. So you have to be at threshold at the hillock, okay? Sometimes called the trigger zone. At the trigger zone, okay? So you have to be at the threshold at the hillock if you want to get an action potential, right? So say we have a sufficient enough stimulus that gets us to our threshold at the axon hillock, right? And so we reach our -55. When that happens, channels called voltage-gated sodium channels open, okay? What do they open in response to? Well, they open in response to the voltage, right? In response to the change in voltage reaching that -55 millivolt threshold, right? And when that happens, if there's sodium channels, sodium channels open, and sodium flows and rushes down its concentration gradient extremely rapidly, right? So if you recall, let's draw a kind of an idealized cell here. Here's a cell. We have lots and lots of sodium on the outside, lots and lots of sodium on the outside with very little sodium on the inside, right? So maybe we'll draw just a little bit of sodium on the inside, okay? So when the sodium channels open, where is the sodium going to flow, right? Well, because sodium goes down its concentration gradient, let's make some little channels here. These sodium channels are going to open, and sodium is going to flow into the cell, right? Into the neuron. So if sodium is flowing into the neuron, and we said earlier that the equilibrium potential for sodium is positive, very positive, that means our action potential, the voltage, is going to move positive, right? So we're going to go from this -70 to -55, and we're going to continue up past 0 somewhere to around +30 or so. All right, now it may not be exactly +30, but it's going to be somewhere around there, maybe +35, something like that. Okay, but the idea is that we're becoming more and more positive. Okay, now after a certain period of time, these sodium channels here are going to close. Okay, so they close off. All right, they close off like this, and now sodium is no longer moving down its concentration gradient, and this is why we see this our graph here over to the left. We see it stop increasing in voltage. Okay, we see it kind of plateau off. Right now, at the same time, we have this. Let me get rid of some of this down here. At the same time, we have our sodium channels closing, or around the same time, we begin to have potassium channels opening. So let's now draw another cell here, and we have potassium. So where is potassium highly concentrated? Potassium is highly concentrated inside the cell, right? So we have lots and lots of potassium here, lots and lots of potassium, and we have very little potassium on the outside. Okay, there's some, but not a whole lot. So when this happens, once we reach this kind of plateau point up here, we have potassium channels open, right? Potassium channels are going to open, and where is potassium going to flow? It's going to flow down its concentration gradient, just like sodium, but in this case, it's going to move out of the cell. And again, when it moves out of the cell, it's going to move the membrane potential closer to its own equilibrium potential, which in this case is very, very negative. So what we'll see is now we have our action potential begin to drop back down. Okay, we see it begin to drop back down. All right, so this phase, let's label some things here. This phase here, let's use we'll just use black. This is our resting membrane potential, right, that we said earlier. This is resting membrane potential. Let me write it a little better here. Resting membrane potential. This is our threshold, so we're just going to abbreviate that TH. This phase here, this is our depolarization phase, right? We're depolarizing. We are moving closer and closer to zero, and we're becoming more and more positive, okay? This phase here, and remember, this has to do with sodium. Let me write this a little bit better. This has to do with sodium influx, okay? Sodium influx to the cell, like we wrote right here, right? This was sodium rushing into the cell very, very rapidly. This next phase, where we're kind of falling here, this is termed the repolarization phase, right? So this is repolarization, and this has to do with potassium efflux, right? In this case, we're looking right here. Potassium is flowing out of the cell very rapidly, okay? So if we are repolarizing, we're reestablishing the polarity. That's what this means, reestablishing the polarity of the cell, okay? So what this means is we're moving closer and closer to that resting membrane potential of -70, okay? But what we actually have happen is we actually move past the resting membrane potential, or the original resting membrane potential, okay? That is because these potassium channels here that open during the repolarization phase are slow to open, and they're also slow to close, okay? So they're open for a longer period of time. So what this means now, if we kind of draw a line going across here, we can see this was our original resting membrane potential. We have now gone under that, right? So we're now below our original resting membrane potential. In other words, we're more negative. So say now we're at like -74 millivolts instead. Okay, we became a little bit more negative, right? And then ultimately we will reestablish this, we'll drift back up, and we'll get back to resting membrane potential until we have the sequence happen again, and then we have another action potential. Okay, so this phase down here, this is called, this phase down here, right? Let me write it kind of like this right here. This is called the hyperpolarization, right? Hyper meaning excess, too much, and then too much polarization. So this, again, this has to do with potassium efflux as well. All right. Now, in order to continue having action potentials, we have to maintain our concentration gradients, right? So we have to keep sodium here. We have to keep sodium on the outside of the cell, like we had right here. We have to keep potassium on the inside of the cell, as we have down here, right? So during our action potential, let's draw just kind of an idealized axon here. So here's an axon, right? We're going to have sodium rush in, right? So we'll have, let's draw sodium in red. Sodium is going to rush into the cell, right? Sodium is going to rush in. And later on, we're going to have potassium rush out, right? So this was repolarization here. This phase was depolarization here, right? So for one action potential, that's fine, right? And 10, that's fine. 100, that's fine. But if we continue to do this, and we continue to put sodium that was on the outside and it moves in, and we continue to take potassium that was on the inside and we move it out, over time, we're going to disrupt and essentially take away that concentration gradient. So now there's more sodium on the inside and there's more potassium on the outside, right? Well, that's not how our action potential works, right? We have to have sodium going in here, and we have to have potassium going out here, right? So what we can have happen in order to reestablish this concentration gradient over time, right? And so one individual action potential isn't going to destroy the concentration gradient. Okay, it would take a lot of many individual action potentials with sodium moving in and potassium moving out to destroy this concentration gradient. Okay, but one way that we can prevent this from happening is by using a protein pump. Okay, and what is this protein pump called? This protein pump is called the sodium-potassium ATPase. So we'll write it down here: the sodium, potassium, ATP ACE, right? So if you recall ACE, anytime you see ACE, this is an enzyme, okay? This is an enzyme that is doing something, okay? Sometimes this is just generally referred to as the sodium-potassium pump, okay? Sodium-potassium pump, all right? This sodium-potassium pump is going to help restore our concentration gradients. So we had sodium that was on the outside and it moved in, and we have potassium that was on the inside and it moved out. We need to put those back. Okay. So what this is going to do is it is going to let's draw another axon down here. Okay. What this is going to do, say now we have lots and lots of sodium on the inside because we've gone through our action potentials, right? We have lots and lots of sodium on the inside, and we have lots and lots of potassium on the outside. Let me do this now. So lots and lots of potassium. Okay, so what this sodium potassium pump is going to do is it's going to take in a 3-to-2 ratio. So let me write this out. In a 3 sodium to 2 potassium ratio, it's going to move 3 sodium. So let's write this over here. Let me extend this axon a little bit. It's going to take, and I'm going to draw the sodium potassium pump as this green structure here. Okay, this is not anatomically correct. Okay, be aware. This is going to take 3 sodium that's on the inside. Okay, so here's 3 sodium molecules, and it's going to pump these back out in exchange for 2 potassium, and it's going to bring these back in. Okay, and by doing this, by reestablishing this concentration gradient of sodium on the outside, let me write this in red, sodium on the outside and potassium on the inside, it allows us to continue to have our action potentials and allows it to continue operating in a functional manner, right? So the big thing here is this 3-to-2 ratio. This is an important value here. And the name of this protein pump, this enzyme, is the sodium-potassium ATPase, right? So if you know it's called the sodium-potassium ATPase, it's probably going to use, let's write it in here, it's going to use ATP to perform this action, and it's actually going to split it and hydrolyze it into ADP and an inorganic phosphate, right? So it's going to use the energy provided by ATP in order to perform this, right? So this is a form here of what we'll call active transport, right? So let's write it right here. This is using active, specifically, let's be specific, primary active transport, primary active transport. Okay, that means that we're using ATP directly, right? We're directly using ATP as we see right here. We're primarily using or directly using ATP to move these electrolytes, move these ions in their respective directions. Okay. So that is everything about the action potential. Please email me. Please reach out to me if you have any questions about any of this, and I will be happy to I'll be happy to answer your question. All right. I will see you in the next video.